Hello, this is Dr. James Camp from Lee College in Baytown, Texas, and this is my second in a series of videos on cell physiology basics, protein synthesis. To protein synthesis. Okay, first off, there are a couple of basic ideas you need to know about how protein synthesis works. The first idea is what we call the central dogma of molecular biology. There are two ways of stating this, or two axioms that are considered dogmatic in molecular biology. Now, you may normally associate the word dogma with religion or politics, somebody who has a firmly held belief that they're not going to change. Well, this is as close as we get to a firmly held and unchanging belief in biology. The first central dogma is that DNA makes RNA and RNA makes protein, or is used to make protein. So we have a DNA structure called a gene. These genes are found in your chromosomes, in your DNA, in your nucleus. They are copied to make RNA, and the RNA that is copied from a DNA gene is called a message, and that message is carried to a protein, and this little machine called a ribosome is used to read the RNA and build a protein from that RNA recipe. The other central dogma is that one gene equals one protein. It's not exactly true, because some proteins have multiple subunits that are coded from a couple of different genes. But the idea is that one gene will never make more than one protein, and usually it's only one or two genes that are needed to make one particular protein. So basically, a gene is a recipe for a protein. So you can think of it as the gene is the recipe or the blueprint, depending on how you want to think about this, for cooking or manufacturing a protein. Why does this protein synthesis business matter to a cell? The answer is that one protein in most cases equals one function. Okay, so you might have a protein that serves as a pump to move things in or out of the cell, or as a gateway to allow things to move in and out of the cell. You might have a different protein that acts as an anchor to keep the cell in place. You might have one protein in a liver cell that stores glucose as glycogen, and another protein that releases glucose from glycogen to fuel the body. Each of those functions requires its own protein, or sometimes a group of proteins, to do them. So we can say, without much exaggeration, that everything a cell does is done by a protein. Therefore, it is critically important for a cell to be able to make the right set of proteins to do all of their jobs. Okay, we're going to talk about how this works, and we're also going to talk about where each thing happens. In the nucleus, DNA is transcribed to make mRNA. That's messenger RNA. There are three kinds of RNA. mRNA is one of them. The DNA gene is read by a molecule called RNA polymerase, an enzyme complex, that matches one base pair of a DNA to one base pair of the mRNA to make an exact mRNA copy of the DNA. Okay, this is kind of like, if you want to think about it this way, DNA recipes are kept on reserve in the nuclear library. And so, if you have a book that's on reserve in the library and you want to be able to use it outside the library, you have to make a copy. So the mRNA message is basically the molecular equivalent of a photocopy of a DNA gene. It's the same language. we use ACGT in DNA and ACGU in RNA, but it's basically the same language. So we call this transcription. Okay, where that word comes from, if you took some notes about this lecture and I wanted to have an exact copy of your notes, I would transcribe them into my notebook. So that's where we get that word. At the ribosome, the mRNA is translated into a polypeptide. Okay, the ribosome reads the mRNA message 3 letters at a time. So you might have A, C, G would equal one particular amino acid, whereas ATG might equal a different amino acid. So the ribosome reads the genetic code to determine the correct sequence of amino acids. So it'll read ACG and it'll pop in that amino acid. Then it'll read TAG and it'll put in a different amino acid next, and it'll keep reading until it reaches the end of the message. And as it goes, it builds one amino acid to another amino acid to another amino acid. And because each of these bonds between amino acids is called a peptide bond, the whole thing is called a polypeptide chain. Why don't we call it a protein yet? Because the protein is the finished product, and we're not quite there yet. We're producing the polypeptide chain that will become the protein. Now, in this case, we're using different languages. We start with the ACGT language of DNA or RNA, and we're working into the 20 amino acid language of peptide chains. So we call this translation. Okay, again, if you had taken notes on this lecture in English and I wanted to make notes in Spanish for a Spanish-speaking student in my class, I would have to translate your notes from one language to another. And that's why we call this translation, is we're translating from the DNA language to the protein language. Okay, so in the nucleus, we transcribed DNA into mRNA. At the ribosome, we translated mRNA into a polypeptide. There are 2 options to proceed from here. In option A, we made our polypeptide in a free or unbound ribosome. We have ribosomes that are just scattered around the cytoplasm, boop, 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 and each one is just going to drop its polypeptide chain into the cytoplasm, and it's going to fold itself into a protein, and then that protein can go off to wherever it's needed in the cell to do its work. This is typically used for relatively simple proteins that they don't need what we call post-processing. They don't need to be modified or specially folded or assembled in certain ways. And it's used for proteins that can do their job anywhere in the cell. They don't need to be specially transported to a particular place. If there's an option A, of course, there's going to be an option B. And the option B is that we have ribosomes on the rough ER. So we've got this membrane here called rough ER, and it's got little ribosomes dotting it here, there, and around. And what they do is that they spit their polypeptides out into the lumen of the ER. The lumen of a biological space is the inside part of that organelle or organ. And in that place, the polypeptide is post-processed. It may need to be folded a particular way. It may need to be modified, have additions or subtractions made to it. It may need to come together with other subunits, and eventually we get something like the finished protein out of that. In option B, typically we then continue to another organelle called the Golgi apparatus. You may remember that, and it's the one that looks sort of like a stack of pancakes somewhere in your cell. And our protein is going to go, it's going to get in a transport vesicle and be carried to the rough ER, to the Golgi apparatus, from the rough ER to the Golgi apparatus. And in the Golgi apparatus, it's going to be finished. There may be some further modification steps we need to do. It's going to be sorted and prepared for shipping. So these proteins that are made in the rough ER often have they have a tag on them that's essentially the protein version of a shipping label. And so they're sorted by where they're supposed to go, and then they may get moved over here, and they get moved over here, and then we get put in another vesicle, okay? And the protein is now carried by a vesicle to one of three places. It can go to a particular spot inside the cell. It can go to the cell membrane itself. Maybe it's one of those cell membrane channel proteins. Or it can go outside the cell. And in that case, we say that the protein is secreted. It is secreted outside the cell. An example of this would be if you were a fibroblast who's making collagen in a tissue, you need to make the collagen inside the cell, but you need to secrete it outside the cell. Or if you're a pancreas cell that's trying to make the hormone insulin, you need to secrete it outside the cell for it to be able to do its job. Okay, so in summary, we have basically 4 steps to making a full finished protein. A DNA gene is used to make an mRNA message. That happens in the nucleus. The mRNA message is used to make a polypeptide chain. That happens in the ribosome. The finished polypeptide chain is then folded and possibly post-processed into a finished protein. That can happen in the cytoplasm, or it can happen in the lumen of the rough ER for more complicated proteins. If we were at the cytoplasm, then we stop there. That there's no need for further processing. If we were in the lumen of the rough ER, we then hop on board a transport vesicle and go to the Golgi apparatus. So we always start in the nucleus. The mRNA message always goes to the ribosome. But then from the ribosome, we have the option of going to the cytoplasm or of going to the lumen of the rough ER. And in the cytoplasm, we're done. Stop. We've got a finished protein. If we're in the lumen of the rough ER, we're going to hop on board a vesicle, go to the Golgi apparatus, hop on board another vesicle, and go to the final destination. All right. And that has been my overview of cellular physiology. I hope you enjoyed it. Thank you for your attention, and happy studying.